I'm here to introduce myself. My name is Adam Jones. I'm a professor at Texas A&M University, and I'm going to talk about simulation-based approaches to understanding the G-matrix. Okay, so what I'm going to do is I'm going to kind of talk about the techniques that we use to simulate the G-matrix, and then the types of things we've learned from these G-matrix simulations. For me, it's been really, really useful because I'm not sure I really had much of an intuitive understanding of the G-matrix and how it behaves in phenotypic space until we embarked upon these simulations. And I think there were some real surprises that weren't immediately obvious from just looking at equations that emerged pretty readily from the simulations that we wouldn't have really comprehended if we hadn't done the simulations. And so this work obviously was done in collaboration with Steve Arnold and Reinhard Berger for the most part. In fact, they're on every single paper that's important from this work. All right, so I'm going to just continue. Okay, so first I'm going to review a couple of concepts, and then I'm going to introduce a couple of other things that you need to understand to grasp these simulations. Okay, so the first one is just the response to selection. So that's already been covered, right, in some detail, and it's come up several different times. And the issue that sort of spurred us to study the G-matrix in the first place was this issue of G-matrix stability, right? So the idea is that it's all well and good to have this equation, and if you can go out and you can measure, the phenotypic mean, that's probably not that much of a challenge. It's probably reasonable to measure selection acting in a particular population at a particular time. But it's pretty laborious to estimate the G-matrix, actually, compared to these other types of things that you need to measure to understand evolutionary change. And so a lot of times we're interested in change for more than just one generation, right? I mean, as evolutionary biologists, we're interested in historical patterns of evolution, and we're interested possibly in predicting the future. And this equation is valid for a given generation, but it only remains valid as long as these variables are the same across those generations, right? And it's completely unrealistic to expect somebody to estimate the G-matrix in multiple successive generations in a natural population or something like that. And so that brought up this controversy about how stable G might be over evolutionary time, because if it's incredibly unstable, then it's not very useful for evolutionary inference. And so that sort of spawned this whole sort of line of reasoning. All right. And so here, this just sort of summarizes sort of the state of the evolution of the G-matrix when we started these studies. So basically, if you compare G-matrices empirically among populations or between different species, you often find some elements of the G-matrix that are relatively constant across those populations, right? So over short evolutionary time frames, there's a fair amount of G-matrix constancy or proportionality. But at higher taxonomic levels, they're quite different, right? So if you start looking between genera or between families, then they become quite different. If you look at very deeply separated lineages, then they're going to be totally different, right? So over long enough evolutionary spans, the G-matrix definitely evolves. And in fact, in some cases, you start to lose traits, right? So you no longer have homologous traits even to compare. And in those cases, obviously, the G-matrix wouldn't even have any meaning for comparison across those different lineages. So at some scale, it does change, right? So that's sort of a fact, right? There's no question about that. But the question is, for the types of things that you want to infer from evolutionary biology, is it so unstable that it can't be used for anything? And that was the kind of thing that we wanted to address. All right, analytical theory neither really guarantees G-matrix stability or G-matrix instability, and it also gives little indication of how much G will change when it's unstable, and particularly in finite populations of the kind that we're dealing with in most natural populations. All right, so the reason we took a simulation-based approach is that these types of approaches had been used, and they had kind of a history in quantitative genetics, and they'd been used successfully to study the maintenance of quantitative genetic variation or additive genetic variation in the univariate case. And so it seemed reasonable to extend these to the multivariate case. So basically, all we're doing is taking univariate models of evolutionary processes and extending them to the multivariate case, which hadn't been addressed at that time. All right, so we wanted to look over long periods of evolution. More recent work, which I'm going to talk about at the end. So I'm going to start out talking about how the G matrix evolves. It turns out that as we go, we're going to see that a big part of that is the M matrix, which is the mutational variance covariance matrix, and I'll define that in a few minutes. The M matrix seems so important to us that a lot of our research has shifted over to understanding the evolution of the M matrix. And so at the end, I'm going to talk about the evolution of the M matrix a little bit, which I think is very, very important. All right, so the model is just a Monte Carlo simulation. So it's an individual-based model where we simulate every individual in the population and every gene in every individual. And the traits are affected by 50 pleiotropic loci. So in this model, every locus is pleiotropic, and it's a two-trait model. So there are always two traits. Every locus can affect both of those traits. In the simple model, it's an additive model with no dominance or epistasis, right? So that makes it a lot easier to deal with the results. The allelic effects are drawn from a bivariate normal distribution with means of 0, variances of 0.05, and a mutational correlation. So this is whenever a new mutation occurs, a mutational correlation of some value that we can specify in the model. So what I'm describing here, this describes the M matrix, right? Which for us, in principle, every locus has its own M matrix, its own mutational matrix. But we're assuming all the loci are identical, right? So we're talking about sort of an average M matrix across these different loci. Here's the mutation rate. We can change that if we want to. Environmental effects are drawn from a bivariate normal distribution with a mean of 0 and a variance of 1. We don't really change that. That's kind of like the scale of the model, right? So you can think of things happening in terms of the environmental standard deviation or the phenotypic standard deviation, which is largely set by the environmental variance that we specify for the model. We have a Gaussian individual selection surface. So that describes, obviously, the relationship between trait values and fitness. Yeah?
So this mutation matrix
For the mutation matrix, what's the dimensions of that? Is that number of loci by number of loci, or is that number of traits by number of traits?
Number of traits by number of traits.
Okay.
So every locus would have its own mutational matrix. So there could, in principle, be 50. If there are 50 loci, there would be 50 mutational matrices. And each one of those would be a trait by trait sized matrix.
Oh, okay.
And on the diagonal would be I'll get to it in a second.
Okay.
But on the diagonal are the mutational variances, and the off diagonal is the mutational covariance, right? So, but in our model, we're assuming that all the loci are the same, and so they all have the same mutational matrix. So there's just one M matrix, which is shared by all the different loci. Oh wait. Okay, so we describe the selection surface with the omega matrix, which you haven't gotten into yet, right, Steve? Okay, so it's basically the inverse. Yes, it's the inverse of the gamma matrix. Okay, so there aren't enough matrices, so we have to have. But the omega matrix, the way to think of it, it just describes the curvature of the selection surface. Smaller values are stronger selection, right? So it's basically the inverse of gamma, which you learned about earlier today. And then we run for some number of generations, something like 10,000, to achieve kind of a mutation selection drift equilibrium. And then at that point, we would start tallying whatever variables we're interested in. Okay. So this is more detail. This slide provides more details on the models. So this is the life cycle. We start with a population of some number of adults, which we can specify. They produce progeny, the mating system is monogamous in this particular manifestation of the program. There's, it follows Mendel's rules for inheritance, right? So, and remember, all the loci are unlinked in this model. So there could be linkage disequilibrium, but there's no actual physical linkage modeled in this model. And then we have mutation and recombination. They produce some number of progeny that we specify. Usually it's 4 per female. There's an equal sex ratio also. We constrain it to have an equal sex ratio. And then there's a selection phase. So this occurs as viability selection in this model. Individuals that survive selection, then there have to be at least n individuals that survive selection. If there aren't, then the simulation run terminates. So we don't allow fluctuating population sizes because that would affect the dynamics of the things that we're trying to measure. Okay, so selection has to be weak enough that the population always stays above a carrying capacity that we specify. And then from those greater than n survivors, we choose n individuals at random to be the next generation of adults, right? So there's a density dependence in here, or a population regulation over here, but it's just strictly truncation, right? So when the population fills up, everybody else just doesn't contribute to the next generation. But there has to be something like that in this kind of model, or you can't control the population size. All right, so in most of the simulations, we start with a population of genetically identical adults. So everybody just has the same allele to begin with, usually a value of 0. And then we let the simulation run for a while to accumulate variation, right? So that's why it has to be a fairly long period of 10,000 generations to get to that mutation selection drift equilibrium. Then we impose some model of movement of the optimum, right? So it might just be stabilizing selection. That's what I'm going to talk about first. Or it could be a moving optimum, right? Which we could specify how it's going to move, and then we just see what happens. And today I'm going to talk about single generation changes in G. So we're talking about changes from one generation to the next, averaged across a large number of generations. Okay, so this slide describes the M matrix, right, which we're going to follow Steve's coloring scheme, and the M matrix is green. And it's just like the G matrix, right? So it's just describing the distribution of new mutations that enter the population. These mutational effects are going to be added to existing mutational effects. So there's some memory in the system. If you already have a large allelic effect and you have a mutation, then the new mutation effect adds to that existing allelic effect, right? So it's not like your former allele is just completely erased and replaced. And then there are variances that describe the magnitude of the mutational effect on trait 1, and variances that describe the magnitude of the mutational effect on trait 2. So those are the diagonal elements. So an M matrix like this you could draw as a circle, right? Because there's no mutational covariance. Over here, it's got the same, this M matrix has the same mutational variances, but the mutational covariance is quite large. So now you have something like this. So what it means, and the mutational covariance or the mutational correlation is very, very, very important, right? So it's critical that you understand that right now, or you're going to be confused for the whole rest. If you're not already confused, you'll be confused for the rest of it. Okay, so the idea, in every single case in this model when there's a mutation, okay, remember, every locus affects 2 traits, right? So both trait effects change whenever there's a mutation, right? But if there's a positive mutational correlation, or a positive mutational covariance, I'm going to use them sort of interchangeably, that means that if the mutation affects trait 1 in a positive manner, then it would also tend to affect trait 2 in a positive manner, right? And so you can imagine that this would be a very powerful way of generating a genetic correlation or a genetic covariance, right? Because if you have a positive mutational correlation, new mutations entering the population immediately contribute to a genetic correlation. But one thing that people don't recognize is that if you have a mutational correlation of 0 or a mutational covariance of 0, even though you have pleiotropy, in the absence of any other factors acting on the population, you will not have genetic correlations, right? So you can have pleiotropy without genetic correlations or genetic covariances, right? Because on average, the pleiotropic effects for this kind of M matrix, on average, those pleiotropic effects are going to cancel out. You'll have just as many positive ones as negative ones, and at the end, you won't have a genetic correlation. So pleiotropy and genetic correlations should not be used interchangeably, right? If somebody says there's pleiotropy, you shouldn't go, oh, your traits are genetically correlated, because it doesn't mean that. Okay. Is everybody okay with the mutation, the M matrix?
Questions. The first one is that the mutation is added at each allele, right?
Yes.
So basically you extract from a normal distribution, you sum the mutational effect to the effect of the allele.
Well, so the mutational effect is drawn from a distribution, is a random number basically drawn from a distribution described by the M matrix.
Yeah, and then you add that to the value of the allele.
Yes.
Yeah.
Yep.
But on what time scale did you observe the genetic correlation between the traits?
Well, every generation. I mean,
So you you found that
yeah.
if you got mutational effects that are correlated, you observe in a reasonable number of generation that it's emerging a correlation between the traits?
in general, yeah. So all else, I mean, you'll see as I go through the results, all else being equal, if you have a mutational correlation, you will have a genetic correlation. Yeah. And it would take time to build up in the population. But in the absence of something like correlational selection, actively removing those relationships that are built up by mutation, you're going to have a genetic correlation. And you're going to have a G matrix that's actually aligned with the M matrix, unless there are other factors acting. Yep.
So is this the only source of genetic correlation in the model, or are there other sources?
No.
Okay.
So the other main source of genetic correlation is selection, right? So the two main things are mutations and correlational selection. All right, as you'll see. So, all right, next slide. So, okay. Go back.
All the things that you said about the G matrix, stability and instability over evolutionary time, is that true also of the M matrix?
Yes, but luckily we can't measure M matrices very well at all, and so you don't really have to worry as much about it because you won't have even one estimate. But yeah, that would be true. Yeah, so if the M matrix is changing over time, and there are a few estimates of M matrices, but it requires a mutation accumulation experiment, which is not something that you can easily do in most taxa. You can only do it in model systems. And it's extremely labor intensive because you have to wait around for mutations to occur, and you have to make assumptions about, which mutations are actually going to pop up in your mutation accumulation lines, and then you have to score the phenotypes of those individuals that have those mutant alleles, right? So basically, that sort of experiment is useful, and it's very, very interesting and informative, but it's beyond the abilities of most of us that work on regular organisms, regular evolution type organisms to do. But yeah, the stability of, if you're going to infer anything directly from the M matrix that involves the M matrix, then you would have to assume that it also is somewhat stable over evolutionary time. Okay. So the selection surface, just briefly, since Steve kind of talked about this already, here it's described by omega. Like I said, larger values indicate weaker selection. Smaller values are stronger selection, so that sort of indicates the steepness of the selection surface. If you have we're only dealing with 2 traits, so it never gets more complicated than this. If you have a 0 for the off diagonals, then it would look like this. It's just a hill, a perfectly symmetrical hill. And if you have a non-zero off-diagonal element, then it could look something like that, right? And so this would be a situation where you have correlational selection, and over here you don't, right? And so this situation, individuals that have large values of trait 1 are going to do better if they also have large values of trait 2, right? This is something you're probably used to thinking about. Okay, and so these are the two main things that we're going to talk about, is correlational selection and the M matrix. All right, so Steve also talked about visualizing the G matrix, right? So there's the G matrix. And then, one way to describe it is with the genetic variances and the covariance. Another way is with the eigenvectors and eigenvalues. If you want, you can draw an ellipse around it. Sometimes I won't even draw that. So we go from the dots to with the cross and the ellipse to just the cross, right? And it's still the same thing. You can imagine an ellipse around it. Okay. There are no questions about that, right? Because we already covered that in enough detail. Are there questions? Question? Okay. Okay. Okay, so in this talk, I'm going to focus mainly on the angle of the G matrix. And the argument that I'm going to use for that, although I'll talk a little bit about the evolution of the variances too, the argument I'm going to use there is that we already knew a fair amount about the evolution of the genetic variances before we even started on the multivariate stuff, right? So sort of the unique part of this study is looking at the orientation of the genetic variation in phenotypic space, which is something you can't do in the univariate case. And so that was already pretty well understood, what maintains genetic variation in these populations. And that definitely is important because it'll affect the rate of evolution for those traits. But in multivariate space, we're more interested in sort of how the standing genetic variation constrains evolutionary trajectories. And that's constrained most by the angle of the G matrix. And so we're going to describe the angle with this Greek letter here, phi or phi or whatever it is. Shouldn't have said that. Now that's going to be on YouTube, but I don't know what that Greek letter is. Right? I can just see the comments at the bottom. There better not be any comments allowed on these things. Is it phi? I did that on purpose. So it's like Steve, I'm testing you. Is it phi? It's phi. Doesn't matter. It's that symbol there. Okay. They'll edit that out. All right. So, all right. So I'm just going to go through some results. So now we're looking at the evolution of the angle of the G matrix. And obviously, I'm not going to go through all the results. I'm just going to hit some highlights. OK, so here's the angle of the G matrix. So it can be from -90. So we're doing it in degrees here. So -90 to 90 in phenotypic space, with 0 being oriented along the x-axis. Right? So this is the angle of the leading eigenvector of the G matrix relative to the x-axis, which is the trait 1 axis in phenotypic space. So here we have a case where there's no correlational selection and there's no mutational correlation. So they're both 0. And you can see that the angle of the G matrix is kind of all over the place. But there are periods of relative stability, right? So occasionally it kind of settles down into a particular orientation. And then that'll be followed by a period of wild instability, right? And then it may settle down again and be wildly unstable again. And then maybe there's a period of hundreds of generations like in here where it's just incredibly unstable. Okay, so that's sort of the result for no selectional correlation and no mutational correlation. There's really nothing here. There's no evolutionary mechanism acting on the population that would introduce additional stability beyond what would just be there from, sort of genetic inertia, a lack of change in the population. All right, and so now with that as sort of the baseline, what happens when we change some of these other parameters? So what we find is that correlational selection actually produces quite a bit more G matrix stability. And so you get a much more stable G matrix if you have strong correlational selection. in the population sizes that we're dealing with, it had to be pretty strong correlational selection to have noticeable effects on the stability of the G matrix. And so you can see now that it does still change. The orientation does still change, but the periods of stability are longer, and the periods of instability are shorter, and it's not quite as unstable, right? And so here I'm plotting every generation, the angle of the G matrix, right? So it's 2,000 points on this graph. All right, so correlational selection increases G matrix stability. If you have stronger, or if you have a strong mutational correlation, we see the same thing. So, but it's much more stable. So here we have a mutational correlation of 0.5. And under those circumstances, we get a very stable G matrix, right? So it just kind of locks in place, and it just stays like that for the entire simulation run. The G matrix may be sort of pulsating and changing in size and wobbling a little bit, but it's not undergoing these really unstable periods like in the case where we have just stabilizing selection or just sort of weak correlational selection. Yeah?
We're looking at a single run here.
Yeah, so this is just a single run here. So these aren't means or anything. This is how stable it is within a run. That's, each one of those is a single run and every single generation. It's not even averaged across generations or anything. That's just the angle of the G matrix generation after generation after generation, right? So that's very, very stable if you think about it.
What's that sort of little pile of dots down at minus 90 there?
So it must have turned all the way up like this. So it must have had a higher, and so it came around. Yeah, and so now it's, instead of being 90, it's now negative 90. And so basically those are the same as, they're just on the top right there. All right. So that's good. So we can get even stronger selection or stronger orientation or stability, though, if the selection matrix and the mutation matrix are aligned. So if we have both a positive mutational correlation and a positive selectional correlation, then again we see very strong stability of the G matrix. And then if it's very, very strong, so if both correlational selection and the mutational correlation are incredibly strong, then it just locks in at exactly 45 degrees, right? So it's just a perfectly stable G matrix that stays stable forever, right? So there are some circumstances under which the G matrix can be incredibly stable over very, very long periods of evolutionary time, but it takes very strong correlational selection and strong mutational correlations. All right, the other thing we observe is that misalignment of those two factors actually causes instability relative to those factors acting alone. So if we have a positive mutational correlation, as I showed on this slide, and a positive selection or positive correlational selection, then we get very strong stability. But if we have a positive mutational correlation and negative correlational selection, then that destabilizes the G matrix again, right? Because they kind of cancel out. And so you can see that on this graph where we have, this is just showing the average per generation change in the angle of the G matrix across a whole bunch of different parameter combinations. This top graph is just with a mutational correlation of 0. And so here it's just showing that as you have a stronger selectional correlation, whether it's positive or negative, you increase stability. But now look at this other graph right here. So this is the case where you have a mutational correlation of 0.25. So here it is with no correlational selection. If you have correlational selection in opposition, so negative correlational selection, then that actually makes the G matrix less stable than it would be with no correlational selection. And if you have positive correlational selection, then it makes it more stable. So when the selectional correlation is aligned with the mutational correlation, then you get more G matrix stability. When they're misaligned, then it destabilizes the G matrix. Does that make sense? Because each force then is trying to stabilize it in a different way, right? So the two different forces are trying to create a G matrix that has an opposite orientation. And depending on the relative strengths of the forces, it can result in, it can sort of crush the G matrix back into a circle, which is the least stable formulation of it. All right, other things that affect G-matrix stability is population size. So these tables just show, here's the change in that angle, the mean change in that angle during a simulation run. This shows the change for 2 different population sizes, and this is a general result. In the larger population, the stability is higher. The mean per generation change is lower in larger populations. And this continues no matter how large the population is. It always makes a slightly more stable G-matrix. The other thing that stabilizes G-matrices is an asymmetrical selection intensity. So if there's very strong selection on one trait and very weak selection on another trait, then the strong selection will compress the G-matrix on that axis, and you'll get an oblong G-matrix that is then quite stable, but it's oriented along one of the trait axes, right? So asymmetrical selection does that. So do unequal mutational variances. So if one mutational variance is much higher than the other, again, that causes an elongate G-matrix. Elongate G-matrices tend to be more stable, and so that also stabilizes the G-matrix. All right, so that kind of goes through all the conclusions for a stationary optimum. So first, I'll just summarize here. Correlational selection increases G-matrix stability, but it takes quite strong selection in many cases to actually get reasonable stability of the G-matrix just from selection. Mutational correlations maintain stability and can produce extremely stable G-matrices, and so mutation seems to be a really powerful way of maintaining these genetic correlations over evolutionary time. And it doesn't have the same costs that are associated with selection, right? In selection, there's a limit to how strong it could be because you have to kill individuals in the population to actually create the G-matrix stability in the first place. That's not necessary for mutational correlations because it happens at the creation of those individuals in their inception. All right. G-matrices are more stable in large populations or with asymmetries in variances, either due to mutation or selection. Alignment of mutation and selection increases stability. This led us to believe that we needed more information on mutational matrices, empirical estimates of mutational matrices, and more theory related to the stability of the M matrix. For some suites of characters, so what I can conclude from this, given all of this stuff, is that for some suites of characters, the G-matrix is probably quite stable. So those that are hardwired to be correlated, so they have shared developmental pathways or something like that, then you may expect those to have a fairly stable G-matrix, right? Because it's built in to the developmental machinery. In those cases, it'll probably be pretty stable over evolutionary time. In cases where it's only created by environmental circumstance, then those sorts of traits are probably going to have less stable G-matrices. All right. So now I'm going to move on and just say a little bit about what happens when the optimum moves. And so I was talking about a case at the beginning where you just had a stationary optimum, and so it's just stabilizing selection, and the population mean stays very close to that optimum. Yeah.
Are you going to talk about what mechanism might be, why the mutation correlation has much more effect than the selection correlation?
I'm not going to talk much beyond what I just said. So if it's because if it's hardwired into the organism as a developmental mechanism, there's sort of no limit to the mutational correlation that you can build into the developmental processes, right? So if you imagine, like, we're symmetrical, right? So we have a very tight genetic correlation between your right side and your left side. And that's sort of built into our development, right? So that can be very, very strong. You can have a genetic correlation that's almost 1, right? If you select for a longer right arm, you're generally going to get a response to selection for a longer left arm. So that's one that's hardwired in there. There's very little cost to that. You don't have to constantly be culling individuals from the population that have dissimilar arm lengths. But if selection is doing it, then the way that it works is you're constantly getting individuals that have dissimilar arm lengths, and selection has to cull those individuals that have mismatched arms and only keep the ones that have matching arms. And that could be a fairly small fraction of the population if you want very strong correlational selection. So there's a very strong cost to the selection because you have to kill all those individuals that aren't right on that line, right on that ridge, the very top of the ridge if you have strong correlational selection. But mutational processes don't have that kind of constraint because you can make traits, I mean, from our own observation, you can have traits that have almost perfect mutational and genetic correlations.
Got it.
It's just a small question. Is your M matrix the same as Wagner's B matrix, or are they different? Are you familiar with that?
Well, I'm not sure.
Because it was
which
pretty
model.
much a map between the genotype and the phenotype. Like,
Yeah, so sort of. I mean, I'm not quite sure which model you're talking about, but I mean, this M matrix is literally just specifying a distribution, so it's probably not exactly the same, but it's just specifying the distribution from which the random numbers are drawn that are the mutational effects.
Yeah.
So it's probably not exactly
I think
the
his
same
model was the effects of each allele on the phenotype. So the mutation matrix is...
This is the effect on the breeding value, right? So it's the mutational effect that's drawn from a bivariate normal distribution. So it simply describes a bivariate normal distribution from which random numbers are drawn whenever there's a mutation. So it's not as profound as those other matrices probably.
So this is, what you're doing is the mutation, the effects of mutation.
Right.
It sounds like you're talking about something that's either the underlying genotype to phenotype map.
Yeah, that's pretty much.
And
But
this would be what happens when you make a change. It's not obvious to me what the connection is. There might be.
Yeah,
it
yeah.
seems-
Actually, I
it
think
seems
he's
to me
right.
that
This
the
is different.
interpretation is pretty similar.
I'm
right?
sorry, I can't. Do you have a micro? Yeah.
Yeah, I have one. Sorry. It seems to me that the interpretation is pretty much the same because you're introducing mutations and they spread to the phenotype.
No,
to the
no, I
breeding
don't think it
value.
is the same because it doesn't. The M matrix has nothing to do with how that underlying genetic value is converted into a phenotype, right? So in this model, it's converted just by, I mean, the conversion.
Except that here, the genetic value adds to the phenotype.
It does.
It's simply
but that's-
additive.
Right,
I
but
think
that's-
there it may be. I'm not sure. I don't know. I don't
No,
know
but-
Wagner's machinery. So
Yeah, but the genotype-phenotype map would be beyond what I'm talking about here with the M matrix. And in this case, the genotype-phenotype map is strictly an additive model, right? So it's just you sum all the loci to get your breeding value. And so it's the simplest possible genotype-phenotype map. Okay. But the M matrix is just describing the change at the level of the genotype, and then whatever map it would be beyond that, which comes up later in the epistasis model, where it's actually an epistatic map from the genotype to the phenotype, but the M matrix is still the same. The interpretation of the mutational effects is different now because there could be a whole bunch of epistatic effects. You get a change in the main effect, then there's a whole bunch of other changes that could occur due to epistasis as well whenever you get this mutation. So then the mutation wouldn't be, the value that's changing wouldn't be the same value that you're drawing as the random number. The actual effect on the phenotype would be something different. And so I think that it's in there that Wagner's thing comes up. Okay, so now back to the moving optimum. Okay, so what happens when the optimum moves? So almost everything that we found, luckily, for the stabilizing selection case still holds for the moving optimum case. But there are a couple of other interesting things that I'm going to talk about now. Okay, so when the optimum moves, so this is sort of depicting what might happen. So say this is the original optimum here, and then it moves over here over some period of time. Maybe it does it instantly, or maybe it takes a while due to environmental change or something like that. And then the population mean evolves toward that new optimum. And along the way, the G matrix is going to play some kind of role in that, right? So it could either, the G matrix could be changing along the way, it could be constraining the trajectory. All sorts of things could be going on, right? And so the question is, what happens? I'm not going to worry that much about the response to selection and exactly how it gets to the optimum. I'm going to talk more about what happens to the G matrix along the way, because Steve will talk about what happens to the optimum tomorrow, I guess. All right. So the most obvious thing is that in the absence of anything else, if there's no selectional correlation or mutational correlation, here's another factor that can increase G matrix stability. So when the optimum moves, that results in a more stable G matrix, even if there's no selection and no mutation. And what happens is the G matrix is just stretched toward the direction that the optimum is moving, right? So it facilitates a response to selection. It stretches out and turns toward the direction the optimum is moving and then evolves along behind it. So it's kind of weird. It's a little bit counterintuitive, actually, because you might actually expect it to be crushed in the direction that the optimum is moving, but actually it's stretched.
But you've got two traits and a single optimum? So the optimum is for just one trait, or it's a bidirectional
No, it's a
optimum?
bivariate optimum.
It's a bidirectional.
So this is in bivariate space. So here's trait 1 here, trait 2 here. The optimum would be a point in bivariate space.
With the same trend? So you got the same.
No, you can move it wherever you want. It doesn't matter. So it could move just along trait 1, and the G matrix would just turn toward that and go that way. It can move that direction so that both traits are moving at the same rate. Both optima are moving at the same rate, and it does the same thing. Or it could just be trait 2 moving. Any of those cases, whichever direction it moves in phenotypic space, the G matrix just turns and goes that direction, like a rat or something. A good analogy.
But what are the red dots in the figure?
Just the population mean or something. No, that's the position of the optimum. Okay, so this is a case where it's moving episodically, maybe.
Actually, the optimum looks like it's changing linearly there. It's a straight line, the red dots.
Yeah, but it depends on the amount of time between them. Yeah, so I don't know. It was just, Steve drew this, and it was just supposed to be a introductory slide. It wasn't supposed to be literal. I'm going to move away from that slide.
Just quickly, you said that the G matrix orients itself towards the new optimum. Isn't that kind of against what, Dolph Schluter said in the lines of least resistance, like adaptation along that it's sort of that it stays oriented in the same way and is in that
and the optimum moves along the
when the optimum the axis
will
not shift.
Right. So Schluter was assuming that you had a stable G matrix over that period of evolution. So if you allow G matrix instability, then it tends to orient toward the direction that the optimum is moving. All right. So So that because it stretches out in the direction that the optimum is moving, then that increases stability. So the other thing that increases stability, and the red dot is the position of the optimum. So it's moving over time some distance every generation or every 10 generations or something like that. It doesn't really matter, it turns out, the amount of time passing between the optimum moving, as long as it stays in front of the G matrix. So the other thing that increases stability is if there's already a genetic line of least resistance, so if there's mutational correlation or correlational selection that gives you an eccentric G matrix that's oriented in phenotypic space, if the optimum moves along that long axis of the G matrix, that increases stability. If it moves across it, it decreases stability. And it makes sense because the movement itself is stretching the G matrix in whatever direction. And if it's going across genetic lines of least resistance, then it's going to tend to get rounder, and a round one is the least stable G matrix. The other thing that's sort of interesting that can happen with a moving optimum is you can get this thing that we call the flying kite effect. And so as the optimum moves and the population mean tracks this moving optimum, if you have very strong genetic correlations, what will happen, so now imagine that only the optimum for trait 1 is moving. Okay, so trait 1 is moving here. Say you have this case where you have a fairly non-eccentric G matrix. It just kind of tracks along behind the optimum. But if you have a very eccentric G matrix, right, that's oriented in some direction in phenotypic space, as this optimum moves, the other trait is actually displaced from its optimum. And so what would happen here, even though only trait 1 is under selection, right, or only the optimum for trait 1 is moving, that, of course, causes very strong selection on trait 1. But if you went out there and measured selection on trait 2, you would see very strong negative selection on trait 2, because that trait has to evolve toward its original optimum because it's being pulled away from its optimum by the genetic correlation. So even though only the optimum for trait 1 is moving, you get the other trait displaced from its optimum, and it could be a considerable distance away from its optimum, right? And so this kind of maladaptation could be especially important as a result of things like global change, climate change, where organisms are evolving toward a rapidly moving optimum, and it's causing them to be displaced from optima at other traits due to genetic correlations, which could have a real cost of selection and population persistence. All right, and then the last thing we addressed in that paper that I'm going to talk about is reconstruction of net beta. So imagine you want to reconstruct the history of selection on your traits, and so you can do that by conducting a retrospective analysis of selection. If you knew G-bar over the whole period of interest, so if you had per generation estimates of the G matrix for the last 10,000 years for your organism, which would be impossible, you could get a really nice estimate of the selection that's acted on that population. If you only have the latest generation, this is the result that we got. And so you can look at this in a positive light, like I would, or you can see negative things about this. But to me, it shows that you can still get a pretty decent estimate of net beta, even only knowing the most recent generation's G matrix. There is definitely some scatter around this, so there would be some error in estimates just based on one G matrix. But in our model, at least, it did a reasonably good job. All right, so we also addressed more realistic models of the movement of the optimum. What's surprising there is that we found almost the same results for a continuously moving optimum. So the first set of results I talked about, the optimum moves just every single generation. Now we address cases where it jumps every 100 generations, 100 times as far each time, or it moves stochastically so that there's a general trend that's the same over time, but every generation there's some stochasticity in the movement. So the optimum would start here, and it would wander around like this over evolutionary time. Or in this case, it just sits there for 100 generations, then it jumps to there, sits there for 100 generations, jumps to there. And so we thought this would have a big effect on G matrix stability. It turns out the effects are pretty subtle compared to just the continuously moving optimum. And so I just want to quickly summarize some of the results from that. So this is just showing some different measures of G matrix stability for a steadily moving optimum. Here's the size, which is just the total, the sum of the two variances. And ignore these two. And then here's the strength of selection, so beta. So with a steadily moving optimum, there's no real, it just kind of goes along, and there's some long-term average, and it kind of deviates a little bit from that over time, and there's autocorrelation, but there's nothing really dramatic happening here. And then the beta is always just a small beta. So you can see it's slightly above 0, right? So you have a small beta that's just always acting on that population. Well, if we have episodic movement, now what happens is beta now shoots up to a really high value, right? Every 100 generations, or this is every 250 generations. It shoots up to a really high value. Then as the population catches up to that optimum, the new location, beta drops, right? Because it catches up to the optimum, so now there's no directional selection. But then the optimum moves again, and so now you get a really strong beta for a few generations while it's catching up to that optimum. You can see that this results, so the main result from an episodically moving optimum is just episodic changes in the total genetic variance, right? So during that initial phase of rapid evolution, there's an increase in the genetic variance, and then it slowly diminishes over time, and then that process repeats itself over and over and over again. And that's pretty much the only new result from an episodically moving optimum. So this graph just shows the same thing with confidence intervals. It's not, but it's basically showing the same thing that was on the previous graph. So I don't want to dwell on that since I'm running out of time. There's a subtle effect of stochastic movement too, right, which you may or may not be able to see here. So if you have steady movement or episodic movement, what I'm drawing here is a case where the only thing affecting the population is peak movement, right? So the only stability conferring aspect of that particular population is the movement of the optimum. And here's what you get for either steady movement or episodic movement. The results look exactly the same. And this is the G matrix under the static optimum. And this is the G matrix for 3 different models of peak movement, where it's either moving up to the right, straight across, or down to the right. And this is the average G matrix that you get as a consequence of that peak movement. So you can see it goes from this round one to something that's oriented in the direction that the peak is moving, right? And it's a little bit bigger because actually directional selection increases genetic variance relative to just stabilizing selection. So here is what happens under stochastic with that stochasticity added. So now it's not quite as effective at orienting the G matrix, right? So you can see that it's not perfectly oriented along the direction that the axis is moving, and that's because sometimes it's not moving that direction, so that makes sense. And then the G matrix is actually slightly larger, so you might not be able to see that, but yet it maintains a little bit more variation. So these G matrices are a little bit larger than these due to the stochasticity. The other thing that happens in a stochastic model is that the effects, when the G matrix is destabilized by the direction of peak movement, those effects are a little bit less, right? So here, when everything's misaligned, you have a really unstable G matrix. Well, that effect is a little bit less in a population with stochastic movement. And when it's really stable, so when everything's aligned and you get a really stable G matrix, then of course it's a little bit less stable when you have stochasticity, because sometimes the direction of peak movement is not aligned with everything else. Okay, and so that's pretty much. Oh, and then the final thing is that both of these things, both episodic and stochastic peak movement, increase the risk of population extinction. So we couldn't necessarily stay above the carrying capacity, and it was a lot easier to stay above the carrying capacity when we had a steadily moving optimum than if we had either an episodically moving optimum or an optimum moving stochastically. And that makes sense because it's just harder for the population to keep up with that moving optimum, right? Because you have these bursts of really strong selection when it's an episodically moving optimum. All right, so the conclusions are that the dynamics of the G matrix under an episodically or stochastically moving optimum are similar to those under a smoothly moving optimum. Strong correlational selection and mutational correlations promote stability. The movement of the optimum along genetic lines of least resistance promotes stability. Alignment of mutation selection on the G matrix increases stability. Movement of the bivariate optimum actually stabilizes the G matrix in the direction that the optimum moves. And both of these other models of peak movement increase the risk of population extinction. All right, are there any questions about that? All right, I'm running out of time. I'm basically out of time. Oh, I have more conclusions, actually. But I wanted to talk about epistasis. So maybe I could just say a few words, like 5 minutes about epistasis.
Quick question.
Okay.
If an optimum moves and then stops moving, moves to a new moves across an area and then reaches a new location, stops. Presumably, this structuring of the G matrix holds for a while. And is it true that once it gets there, it might return to to mimicking the?
That's exactly right. Yeah. So it'll stretch out while it's going there, and then it gets there, and it would go back to whatever it was before it started to move. All right, so a few other conclusions from the main part of the talk. Episodic movement results in cycles in the additive genetic variance, the eccentricity of the G matrix, and the stability of the angle. Stochastic movement sort of tempers the effects of stabilizing and destabilizing effects. stochastic movement actually increases additive genetic variance. Actually, selection, so this is something I didn't talk about, but it's in the paper. So selection actually skews the phenotypic distribution, which you assume to be normal when you do selection analysis. But actually, it's non-normal due to selection, which actually changes the response to selection. And so if you care about that, it means you could be wrong by a lot, by like 15%. But most people don't care about a mere 15%, I guess, for some reason. But it actually, the selection itself changes the phenotypic distribution in a way that affects the response to selection. And so that's something you may want to think about if you're really interested in the response to selection. All right, and there are lots of other things that could be addressed with these simulation-based models. Okay, so for the last part, is it okay if I keep going for like a few minutes? Okay, all right. So the last part I want to talk about, which will hopefully wake everybody up now, is epistasis. So everybody's worried about epistasis, right? And that's with good reason, I think. And so I think that epistasis is probably quite common. I mean, everything that we know from molecular biology and developmental biology indicates that gene interactions are quite important. And so what we wanted to do, and the reason that we sort of got into epistasis came from the evolution of the M matrix. And so as we started doing these studies, I think we expected selection to be sort of the most important thing. And we were sort of surprised by how important the M matrix seemed to be. And in retrospect, it makes perfect sense. But then you start wondering, well, why do we have an M matrix that is what it is? You know, why is there a genetic correlation, or why is there a mutational correlation for these traits and maybe not for these other traits, right? So the question is, what shapes the M matrix? And so we started to wonder if maybe selection could be important in determining the M matrix in the first place, right? So if that's the case, then maybe you get an M matrix that's more favorable for a particular environment as a consequence of selection, which is a different way of thinking about it than people normally think about it, right? So when you're in intro biology or intro evolutionary biology, you learn about mutation, and that's one factor. And then you go, okay, well, that's where the raw genetic material comes from. And then you learn about selection separately. And you go, oh, geez, I hope there's enough mutations that I have enough variation upon which selection can operate. And you sort of treat them as being completely separate. But in fact, it turns out that selection can affect the nature of mutations that are entering the population in the first place. All right, and so we studied this in a paper that I'm not going to talk about at all, where we just focused on the evolution of the mutational correlation itself and sort of treated that as a quantitative trait. And we found that selection does act on the mutational correlation. Selection is weak, but over long evolutionary periods, you can actually see a meaningful response to selection in that mutational correlation. But that model was contrived, kind of, because probably mutational correlations aren't just a quantitative trait, right, that can be modeled with 10 additive loci. A more realistic way to model this is with epistasis. So epistasis actually provides a way for the M matrix itself to evolve without having to assume mutational correlations in that underlying genetic architecture or mutational architecture. And so we wanted to ask, well, what will happen if this kind of selection that we see on the mutational correlation acts on a system that has epistasis? Okay, and so and we have a paper that just came out this year in Nature Communications on this, if you're interested in reading about it. All right, so the model is Hansen and Wagner's multilinear model. And so it's just an extension of the additive model. It's a fairly simple extension, really. And in fact, adding the model to the simulation isn't that hard. All you do is you add these other terms, right, the epistatic terms. And so it's just like the additive model where the phenotype is the sum of all these additive effects plus, an environmental effect. And now you still have the additive effects, but now you have epistatic interactions. So these are loci. So locus 1, locus 2, locus 3. So now you have locus 1, locus 2, and then you would have an interaction between those two loci with an epistatic coefficient that tells you the magnitude of that interaction, right? The problem is with 50 loci, you have literally hundreds of these interaction terms, right? And so we actually modeled it with 20 loci, but you still have literally hundreds of the interaction terms. But basically, that's the model, so it's not that hard. You know, it's not that much more complicated than the additive model. And for the multivariate case, it's a little bit more complicated, but I'm not going to show that up here. But if you're interested, it's in the paper, and so you can read about it in there. But the question is, what happens when we add epistasis? And I'm not going to go into all the little details. I'm just going to go quickly through this because I knew I'd be running out of time. OK, so one question is, does epistasis increase genetic variation? And the answer is, you would expect it to, right? At least the total genetic variation. And the answer is it does sometimes, but it's somewhat complicated. It kind of depends on other factors. But in many cases, it does. And when that happens, a lot of times there's an increase. As there's an increase in genetic variation, so this is the value of those epistatic coefficients, right? So this ranges from no epistasis, so that's just the additive model, to very, very strong epistasis, right? So the reality is probably somewhere between 0 and 1 here, although we don't really know. But what happens is as epistasis gets stronger, then you get the percentage of the variation that's additive diminishes. But there's still a fairly large amount of additive genetic variation in this model. No matter how much epistasis we add, there's still a lot of additive genetic variance. All right, so that's one thing that happens in the model. Another thing that happens is that the genetic correlation evolves. And what's interesting is that so here's a case where there's selectional correlation. So what's interesting is the total genetic correlation. So if you look at the total variance, so this isn't the G matrix, this is just total genetic variance. The genetic correlation actually decreases. But if you look at just the additive genetic variance, the additive genetic correlation is actually sort of increasing, although it eventually, also it's a nonlinear relationship between that and the strength of epistasis. Right? So the overall genetic variance could be quite different than the actual additive. I mean, this is the genetic correlation. The actual additive genetic correlation could actually be quite a bit stronger than the observed genetic correlation that you would get just by looking at the total genetic variance. The more interesting result from our perspective is that we see evolution of the mutational architecture. So M1 and M2 are the mutational variances for the two traits, right? So this is the variance of that distribution from which the values are chosen. The underlying parameter for the mutational effects that are drawn from that bivariate normal distribution stays the same. So the variances are 0.05, right, for the main effects. This is the observed effect of the mutations after all that epistasis is factored in. And so what we see is that in small populations, we evolve very large mutational variances. And in large populations, we evolve very, very small mutational variances. So there's the evolution of the mutational variance seems to be affected by population size, which we interpret as being an effect of selection for smaller mutational variances for large populations, which tend to be close to their optimum, right? This is a case with stabilizing selection here. And then the other thing we see is that the mutational correlation does evolve. And so if we have larger populations, and here we have correlational selection, really strong correlational selection, in those large populations, we evolve a positive mutational correlation. The underlying main effects are still being drawn from a distribution that has no mutational correlation. This is the mutational correlation due to the epistatic effects. Right? So epistasis causes a positive mutational correlation to evolve. So epistasis does allow the mutational matrix to evolve. All right, if it doesn't make any sense right now, I have some figures that'll make more sense in a minute. So one of the results of this model that's sort of counterintuitive is that in the strictly additive model, which is shown as these red diamonds right here, the genetic variation increases with population size, the additive genetic variation, right? And this kind of makes sense. In the model with epistasis, we see a situation where the additive genetic variance actually decreases with increasing population size, with larger populations having less genetic variation when there's reasonably strong epistasis. This is the value of the epistatic coefficients here. And there's sort of a nonlinear effect, because if the population gets too small, then suddenly you start to lose variation due to really strong genetic drift, right? The reason for this is shown on the next slide. So the reason is the evolution of the mutational variance. So in the small populations, you evolve very, very high mutational variances, and that introduces a lot of variation every generation into the population. In the larger populations, the mutational variances evolve to be quite small, so mutational effects are small, and so whenever a mutation occurs, it doesn't introduce that much variance, and so you maintain less additive genetic variance in those larger populations. All right, the other thing that happens in this model is that everything ends up being aligned, right? So if we have the selection surface, the G matrix, and the M matrix now, we can plot those in phenotypic space and draw those ellipses around them. And so we see in this case where there are zeros for all the off-diagonal elements with strong selection on trait 1 and weak selection on trait 2, the selection surface is shaped like this. The G matrix is sort of nested within that, and the M matrix is nested within that. We can rotate that through phenotypic space, right, and see what happens for simulations run under different orientations of the selection surface. And what we find is that in every case, the G matrix, this is a case where it actually orients really well because it's a very large population size, but the G matrix orients toward the direction of the selection surface, and then the M matrix also orients toward the long axis of the selection surface. So the M matrix evolves to be oriented in phenotypic space in along the same axis as whatever the selection surface is in this model. And then here's another example. And so, and these are from real data from the simulations. It's just really good alignment. But it's an average across a long run. And so it might wobble a little bit, but on average, it's oriented with the selection surface. All right. So the conclusions for epistasis, or that epistasis allows natural selection to shape the mutational architectures of traits. So I think the mutational architecture is partially a product of natural selection. It's not just something that arises, magically from mutational constraints or developmental constraints. Those are being shaped also by natural selection. Smaller populations evolve larger mutational variances. Smaller populations in this model sometimes harbor more genetic variance than large populations. And then we get this triple alignment where the selection surface, however that's oriented, everything else evolves to be oriented in the same direction as the selection surface. Of course, that's assuming that it's a fairly stable selection surface over the simulation run. All right. And so the implications of that, this is the last slide. Yeah, okay. Are that the first really big implication that was sort of eye-opening for this is that just because you have a lot of additive genetic variance doesn't mean that there's no epistasis. You can't go, oh, I have really high heritabilities, so therefore I have a whole bunch of independently acting additive loci. Much additive genetic variance arises from the epistatic effects. So epistasis also causes additive genetic variance. So this idea that people have that there's heritabilities are pretty high, so there must not be any epistasis. That's just not a reasonable way to look at it because a lot of additive genetic variance can arise due to the epistatic effects. So those interaction terms are contributing to additive genetic variance as well. They do contribute some non-additive genetic variance, but they contribute additive genetic variance. Mutational variances and covariances can be shaped by selection when epistasis is involved, and so that was the main thing we were trying to explore, and we found that indeed that's true. There could be hundreds or thousands of these epistatic interactions in real systems for highly polygenic traits, and so it could be very difficult to actually find them in the genome and estimate the magnitudes of their effects. So that's a real constraint that's probably present in natural systems. And the final conclusion is that the mutational architecture, which we sort of learn is separate from natural selection, is probably actually shaped by natural selection to begin with. And so, all right, thanks for bearing with me and going while I went way over time. So, thanks.